All living beings depend on photosynthesis. No escape. Oxygen, food, and I'm saying whatever we read about it in textbook, in this is all wrong. And it's not wrong by kuti piece or anything, totally wrong. Okay. So uh, and I'm very confident that it is wrong. My model need not be, and by the time you leave this room, I'm pretty sure that you'll also be very confident that it is wrong. Because wrong things, they call out very easy. Right things, you have to have a lot of information to say this is right. So in the light reaction, I'm focusing only on the light reaction. We need to account for the formation of ATP, NADPH, the reducing equivalents, and how the oxygen is liberated. Then the current mechanism is highly vitally deterministic. Okay. Now, I'll just go quickly on the structure of chloroplast. Do you know all these structures? Basically, the grana, stroma, all that. You might have studied. Some of you might have studied in school. I think I'll not go there. This is the best place. So, yeah. So here, these are thylakoids, flat lipid membrane, and photosystem 2, photosystem 1, light harvesting complexes, blah, 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 all that are present here. Okay, I'll not go into details. Now let us, the usual cycle, usual exercise. First, chloroperoxidase cytochrome P50, photosynthesis, respiration. Now I'm doing respiration and photosynthesis system comparison. What I'm saying is, if one system has some mechanism, another system with similar components, it's highly unlikely that it will not have the same mechanism. It's not like, one is in, one system says 2 + 2 is 3, another is 2 + 2 is 4. It cannot be like. So that's the point. So mitochondrial oxidative phosphorylation system, photolysis photophosphorylation system. This is chloroplast. This is mitochondrial oxidative phosphorylation. If you see the so-called electron transport chain, the arrangement, they are same, but reverse direction. In one, you have oxygen giving water. In the other, you have water giving oxygen. Right? They are anti-parallel system. All right. Now, but then that is made possible because of the energy of light that is input. Now, you see this. Now, what I've shown here is, in everything, there is a very clear comparison. 2 electron into 2, everything. So basically, it's very much, some are proton pumps and all that, very similar. And there is a complete comparison, pan-systemic comparison. That is, in microsomal xenobiotic metabolism, photolysis, photophosphorylation, mitochondrial oxidative phosphorylation, all of them are this coupling, uncoupling, and oxygen, all these nicotinamides, all that, processes. So basically, the mechanisms involved have the same logic. I'm trying to make a superficial bird's eye view. Okay, please be with me. This is very important. Even if I could understand, so can you. Now here, oxygen evolution. This was a turning point in photosynthesis research. It's called Emerson effect. What our Illinois, Urbana-Champaign people. So here, 680 nanometer is photosystem 1. All right, 700 nanometer is photosystem 2. When you excite those photosystems separately, you get this much oxygen. All right, but you excite them combined together, what you get as individual and added up, it's much more than that. Did you get that part? Okay, that's one. So what they did is, they said that they work in these photosystem 1 and 2 work as a series of electron transport system. That is, they work in series. You have got two, either, I'll come to that analogy later. So this is called the Z-scheme ETC. This is the paradigm that explains photosynthesis in textbooks and all research endeavors. Now, so serial modality, 2 molecules of NADP held at reductase, 4 electrons. Yeah, sure. So at photosystem 2. So yeah, so this also has the electron transport chain, and you have the electrons coming from water to going to plastoquinone 2 to cytochrome B6F to plastocyanin to photosystem 1 to ferredoxin and reductase, giving NADPH. All right. This is the electron transport chain, which is practically the reverse of that. But you can see plastocyanin can be compared to cytochrome C, plastoquinol can be compared to ubiquinol, all that. It's very similar. So, and the photosystem 2 was earlier supposed to be a proton pump. They conveniently changed it later. Oh, this is not a proton pump. You can imagine, if it is a proton pump, it should be easily ascertainable. It's a fulcrum of the theory. Peter Mitchell said that complex 4 was not a proton pump. And the followers said, no, sir, they are proton. No, it's not a proton. Mazaka, yeah. It's a fundamental part of your theory, and you say it's yes or no. It's not game, but they played games. Here, they changed again. Okay, plastosystem 2, photosystem 2 was supposed to be a proton pump. It became a proton, it didn't hold that status anymore. So then the Kok-Joliot cycle. Kok-Joliot cycle was the mechanism for oxygen evolution in manganese complex of photosystem 2. Cyclic charge relay transfer and all that. I will not go into the details because it's already late here. So, but then the point is that what it shows is that 2 molecules of water were supposed to be held at manganese complex, and they were systematically, electrons were released as light impinged upon photosystem reaction center of photosystem 1 and 2 located elsewhere. And these electrons were systematically ferried across the whole point like this to finally make NADPH. So basically, what it says is that it must make several dozens of electron transfers to make 1 molecule of NADPH. Do you understand? Evolution had to go through several dozens of steps to make 1 molecule of NADPH, which can be easily made at 1 step. Why would it make? Because to explain this Emerson effect, they made the arrangement. So this is Q cycle for plastoquinone regeneration. Okay, I'm not going into the details. I'm not going. This is the complex 2, complex 5, which was supposed to be the protein ATP synthesis, harnessing transmembrane potential. So quantum computing light harvesting complexes. So you've got a random distribution of light harvesting complexes, which was supposed to collect photons and then relay it as excitons to the photosystem's reaction centers. So how does it do it? How can anything do it like that? Without any energy loss, it's supposed to relay. I don't know. Very sophisticated logic. I don't understand. Let me criticize. I like this part. Now, you have 2 resistances. You help me out. You people are the people here. Whether you can consider it as resistance or capacitance, right? Now, there are electron donating or they move. Let us say electron transport chain is like a circuitry. So you have current flow, yes? Current flow or say fluid flow. You can equate it. So now if you take photosystem 1 and photosystem 2 as 2 pipelines or 2 circuitries or 2 capacitances or 2 resistances, in whichever way you want to take, they are made in series to explain the extra yield. Do you think series will explain extra yield or parallel will give extra yield? Parallel. It's basic thing. Now, traffic from point A to B restricted. You want to increase traffic now, basically. So what do you do? Put more, let go more, parallel, very simple. Even if you invoke a capacitance, even if you invoke a reservoir, it makes sense in any computational, in any analytical purview. This system has to be parallel connection. They went for serial. Sad. Okay, and the next part. Plastoquinone is supposed to relay electrons from photosystem to cytochrome B6. Now the problem is photosystem 2 is located deep inside the ground. So plastosystem 2 should say I should jump lipid membranes from one stack to another stack. Just move. That's what is happening here. I feel sorry, but this is not possible. So these are all, I don't know why they came up with such ideas, but anyway. And how can all these plastocyanin, you knock out plastocyanin completely. Nothing happens to the plant. It grows fine. It photosynthesizes fine. And that was an experiment done in 1990s. And for their theory, if you see, इसके ऊपर अटका है पूरा इलेक्ट्रॉन एक one single thing. If this thing goes, circuitry broken. There is no electron flow. So this thing, you take it out, still it works. And then the topography. If it was, this is the crux of my theory. If this is such an important electron relay, what would you expect? Its topography should be maintained. It doesn't. And it's only at 1 micron. You want something to relay quickly. It's a diffusible agent. You will want it in higher concentration. It's practically. In my theory, it is that, okay, in the cyanobacterial system, cyanobacteria is one unique system where you have photosynthesis and respiration occurring simultaneously on the same membrane. Now you see this, the electron transport chain. It has got the cytochrome C. Now, if it goes to if it is going from the photosynthesis part to the next photosystem 1, then it takes the electron to NADPH. But if it takes from to complex 4, it is making water. So how does the cytochrome C know? I'm doing photosynthesis or am I supposed to do respiration? Which way do I go? How does it make the choice? Did you understand my point? It doesn't have any logic. It cannot work. It works by a electron transport chain. This is, I think I should make a solid mockery. Otherwise, these people will not change. The man who made the cyclic and acyclic photosynthesis, that man, when he proposed, he showed in photosystem 2 itself, NADPH is getting made here. Why do you need such a long circuitous way? It's a very simple thing. Electrons come out and NADP gets reduced. Arnaud, Daniel Arnaud. Have you ever come across this name? Daniel Arnaud. He made the cyclic photosynthesis, acyclic photosynthesis. That one. So poor man, they all silenced him. Unionism. Sad. I like physics for that matter. You cannot say these kind of things and get away in physics. I want this to be recorded, particularly after watching. So here I have systematically computed every single step as per the contemporary theory. You see here, in terms of kinetics and thermodynamics, the same elephants and Marty's story. One-to-one correlation. There is one thing to take it to another, but then Does it explain the kinetics? Does it explain the process? No. So this every single step of the photosynthesis, the 19 steps that they say is required to make from water one molecule of NADPH. Can you imagine? Can you imagine any reaction scheme where to make one molecule of 2 electron reduced NADP to get NADPH, it requires 19 steps. Will it enhance the rate? Will it enhance the yield? It will only mess things further. That's what is in the textbook. So, Kok-Joliot experiments that led to cyclic analysis. Sadly, the data given by the pioneer I analyzed, that the claims by our University of Illinois website, that itself is questionable. That direct, straightforward interpretation of data, that itself. And then recent data, all they think is they thought that it's a 4 electron reaction, right? 2 water molecules releasing 1 oxygen. So it's a 4 electron reaction. So it has to go through by a systematic way. And so they said they have 2 states of intermediates. One is 1 electron deficient and another is 4 electron deficient. And so you will get in one, after 3 steps, you will get oxygen evolution, and then in one you will get. So that is the ideal one. But and they say that this is the person who modeled. You see, they are getting 10, 1 ideal approach. But then the other data sets by other groups, they don't conform to the requirement. So the that is not conforming. Then the data bicarbonate was shown to enhance oxygen evolution. The classical Z scheme cannot explain how bicarbonate enhances oxygen. So here, bicarbonate, you see, from bicarbonate, oxygen also comes. All these things. Then how can, in the classical theory, only photosystem 2 is capable of releasing oxygen? Then why did Emerson find with the photosystem 1 also oxygen evolution? Why? No. So it's a highly deterministic scheme. It has to be highly coordinated. I'm not going into the detail of this. And it requires all these. What I am saying is analyzing that if 2 molecules of water are held at one place, all the other system components have to be in a definite arrangement. If they have to work in a series in a particular way, and they have to work in such a concerted way, it's difficult. And the electrons have to go like that. But then the structures have to be very beautifully placed and in very beautifully in a redox state, which is not the case. That's what I'm saying. And the coenzyme Q cycle, every single step I have analyzed that. Once again, it's not possible. So this is beautiful. This is the there was a paper in PNAS. Free energy landscape is conserved, they say, of the Q cycle proteins. This is the Q cycle proteins. Where is the conservation? Where is the conservation? There are redox centers everywhere here and there. How does the electron know from here to there I go? It doesn't know. It cannot. There is no deterministic thing there. And now I introduced a simple methodology to assess probability. You say binding on and off, you give it half probability. Binding on and off. And then you analyze one mechanism for the outcome probability. So if with this Q cycle, simple Q cycle that Peter Mitchell proposed, this probability score is something like 1 in 1,000. How can it conduct like that? The quinone head group changes structure. So it's not a binding specific reaction. So this is what you see in your textbooks. Jagendorf experiment. Do you remember Jagendorf's experiment? What they did is equilibrated chloroplast at 4 pH, and then elevated the external uh, the environment pH. So they found ATP. Array, what is the great thing in that? The physiological reaction is not like that. Physiological reaction, chloroplast, inside, outside, it's 8 pH. How can you do this and show that that is the reason the pH gradient in whether it is ATP synthesis or whether it is NADP reduction, you have proton requirement. That is ATP plus H2O gives ADP plus Pi plus H plus, and NADP plus H plus gives NADPH, that 2 electrons. So you put H plus into that system, you will get. How can you show that pH gradient is what is driving the ATP synthesis? That is not the physiological demonstration. And then Racker's experiment. What Racker did is to complex 5 and rhodopsin. Sadly, rhodopsin is not a proton pump. They thought rhodopsin was a proton pump. Rhodopsin is an electron emitter. It has got a carotenoid, which is what I will talk about tomorrow vision. So it generates ROS. And your question, no, what complex 5? That is a function. So basically, rhodopsis pKs keeps their base pKs 13. 13. What will you expect in pH? This thing, it doesn't. It is a ROS producing system. So wrong experiments were misinterpreted and all that. And then it cannot explain the action spectrum also. Basically, because there is significant photosynthesis in this region. So, yeah. Now, if you this is wonderful. You see this. Now, this is supposed to be maintained at a fixed polarity. You see Z skin. Now, here ions are supposed to go in this electrochemical system in a particular direction. Here, electron goes, proton comes in, proton goes, electron comes in, electron goes. What is the potential point of maintaining a potential across a membrane? If ions can come in and go out, what is driving that ion to move in and go out? Will that potential not see? Potential is generated because of ions move, or ions move to maintain the potential. Here, which law of physics is it abiding by? Which law of chemistry is it abiding? Nothing. And you see the textbook explanation. 2H2O gives reacts with NADP, gives this thing, Z scheme etc. And the Chemiosmotic rotary ATP synthesis, the crash is this. The total yield is 1464 kilojoules per mole. That much is needed. 4 photons of 680 nanometers and 4 photons of 700 nanometers amount only to 1387. Where is the extra energy coming from? This is given in the textbook, not said by me. So if this is wrong, photochemically alter them. So basically, here what you have is multiple systems have to come together, cyclic system. What kind of cycle this is, I don't understand. And when you see the Z scheme, you see conveniently 680 to 680 star. But then the point is, electron cannot go like that. Excited state, then the electron deficient state, and the neutral state. There are 3 states. Take and give name. There is another state. So when you draw, it also has to go to a cyclic state. So all these things to act in a synchronized way, it cannot. The Murburn model is very simple. It says that I'm not going into this. Electrons can come and go in multiple ways. This is the thing that explains the parallel arrangement. There's a reservoir, the photoactive pigments. In my model, light harvesting complexes don't do quantum computing, don't transfer excitons to the reaction center and all that. Now you imagine there is oil in the engine, right? And then there is fuel to burn, right? The fuel to burn is the one photosystem produces. Light harvesting complexes are also producing ROS, but they are like They are like enabling the smooth movement of the engine. That is, they are like lubricating the system. They are like oil. They don't get burned. So light harvesting complexes, when these systems get electron deficient, the light harvesting complexes give their, but they are not efficient charge separators. So on their own, they are not capable. With light harvesting, with photosystem 1 and photosystem 2, they efficiently separate charge. So you have what I said. The whole concept is about moving electrons and moving moieties. Effective charge separation is a fundamental premise. So you provide energy, and I have here, this is a primary event. Diverse pigments, photosystem 2, photosystem 1. Electron emission occurs, giving DRS and electron. Diverse pigments, then electron replenishment occurs. All these that become deficient, they must be replenished, and that replenishing is by DRS again. And then you have NADP taking the electron to give this, and then the DRS and all that reacting with ATP to give phosphorylation. Very simple, delocalized reaction. Did you understand the scheme of photosynthesis? Now I will. So let us go. These are the equations. Basically, you see the equations. Very simple bimolecular equations. Support for Murburn model. You see this chlorophyll, carotenoid. They have to randomly distributed. That's the max. The major picture. There is no binding of the chlorophyll to a particular part, and there is no genetic determination of a structure or an arrangement. It's a stochastic scheme. There is no escaping that. That's what it is. So, and then the matrix or the non-membrane part have ADP binding sites. So ATP synthesis also ROS produced attacks. So what are the support? That is, such diverse molecules serve as electron donors and electron acceptors to photosystem 1 and 2. If it was a specific one, how would you expect it? It's not. So it can also explain bicarbonate-based oxygen evolution by simple ROS-based chemistry, wherein the oxygen can go into water formation without coming into the oxygen, and it can also give in carbon dioxide also. So paradigm shift is we have to discard Z-steam, Kok-Joliot cycle, Q cycle, etc. Amorphous arrays can give effective charge separation without invoking any intelligent quantum computing. Reorientation of bioenergetics is complete. Cellular bioenergetics, which powers cells. There are two fundamental routines that power cells. One is respiration, one is photosynthesis. I presented today, the photosynthesis was not that effectively presented because I was trying to make up time for the students to have their lunch. So here I wanted to thank Afsar Manikatudi and my Chinese academic sciences collaborator and you who collaborated with me for the bicarbonate part. So yeah, so this is it. So cells need powering, and the two mechanisms that are used for powering both are absolutely wrong in the textbooks. And that has to be changed. We cannot teach this any further. I don't care whether my model is taught, but I really care that what we're teaching now is not taught. And I don't need anybody's endorsement for that, because the wrong thing is something that can be said by anybody. And I am not going to wait for people to claim that it is wrong. It has been demonstrated. Whether my theory is correct or not, that I don't know, because I've just proposed something. I haven't conclusively demonstrated anything. But given the fact that I've demonstrated in 2-3 different systems with similar components, it is obvious. Only difference in photosynthesis is one step, that is the involvement of light. And that part, even in photodynamic therapy, in humans, you apply red light, we also make ATP. It's the loss. Some aphids which have incorporated some genes from some plants, they have green parts in their body. They also make ATP. They also make NADPH. They do photosynthesis, but they don't have electron transport chain and all these all these things. They don't need all these sophisticated ideas to make simple chemical reactions in a stochastic ambience. The perception was highly ingrained, highly entrenched. Some senior people determined what is going to be what. It's no problem, but when you bring new information, when, for example, when plastocyanin, it was shown plastocyanin oblique, you knock out plastocyanin, and when it was in 1990s, what is it being taught? Who is it being taught to our younger generation, even in 2020? How can it be? I'm very, very upset. I'm very upset with people who are doing this research. It's not fair. That's not what we are. That's not what humanity is about. Humanity is not about keeping things intact for a few people. No, it's not about that. And I want it to be documented. Yeah. Yeah. So anything else? Anybody from the group is asking anything?
No.
No.
So we will be available for personal interaction from.
If you want,
yeah,
I'm here.
3 p.m. in the room right across 303.
Anybody will be coming? Because if you are coming, only then I need to be here. Otherwise, I have 2 papers to prove. I'd rather prove my papers. So if somebody is coming, even if one person is coming, I will be here. So, but don't feel obliged or pressured just because you thought that this guy is a passionate guy. Let's no, no, don't do that, because
You'll be there tomorrow.
Yeah, I'm there tomorrow.
If you have a question, you can. Yeah, you can just because there's a lack of time, no? So you'll be there tomorrow.
Yeah.
We want.
No, that's good. That's good. Be honest, please. I'm quite happy.
So let's all thank Murali for a really exciting session.
Oh, at least a few of you turn up tomorrow.
Yes.
And he's open to pressing questions.
Yes.
He's an advocate. So feel free to ask.
Free. Thanks.
Thank you to the audience.
So this part,